this presentation is a little bit different flavor compared to the previous presentation, which was very in-depth and rigorous. This is more of a high-level introduction to mass spec and how it is actually used for proteomics, lipidomics, metabolomics. Hopefully, by the end of this, you'll come away thinking that it's very simple and you'll know exactly how to use it in your own research, right? So what are the MS-based omics? Primarily, they're non-sequencing type molecules, right? So this is basically proteins, metabolites, lipids, things that can be turned into small molecules. So why would you use mass spec typed omics? Basically, it's going to be the phenotypes that are dependent on non-genomic information. For example, nutrients, toxins, proteins like structural proteins, these types of things aren't really readily captured by sequencing type technologies. So this is where it really shines. Some potential relevance for chicken science, basically, you can look at meat quality, you can look at animal health and welfare, for example, stress markers like cortisol. You can look at nutrients, right, which are not readily measured by any other methods. And then even in some cases, you can look at microbiomic contributions to plasma, and then you can see different colonization. So how does it actually work? Hopefully, I'll demystify it a little bit. Basically, there's one main mode of doing mass spec to analyze all of these different types of molecules. It's LC-ESI-MS. That acronym is basically liquid chromatography, electrospray ionization, and mass spectrometry. After that, you do quantification and identification. It's going to be a little bit shuffled here, but I'm going to start with mass spec. This technology is really, really space age, in my opinion, but it all comes down to measuring the intensity of different masses, right? And it's really based on two main principles. One is you're using electric fields to control ions or charged particles. Here you can see an animation of some charged particles just being juggled by alternating electric fields. This is what the modern mass specs do. They just change electric fields and push ions around. The second principle that's used in mass spectrometry is that larger masses tend to have more inertia, so the smaller molecules tend to travel faster than the large molecules. So one of the simplest ways to measure mass is a time-of-flight mass spectrometer. You basically get some ions in, you pulse with a voltage this way, and they fly towards the end. There's a detector on the other end. Depending on how long it takes for those ions to hit this detector after that really short pulse, you can determine the mass, or mass to charge, really. And then, depending on the signal that comes back from the detector, you can measure the intensity. So you can see just these two main principles can be used to measure different masses. But how do you generate ions? Well, there was a key breakthrough. Basically, it won the Nobel Prize in the 2000s. Basically, you apply a high voltage to a capillary, a small tube with your solution, and it will just spontaneously spray and form ions. And so you can put a mass spec at the other end of the volts, and then the ions fly into the mass spec. I mentioned this specifically because the mechanism of ESI is that in most cases you're adding or losing a proton, so any molecules that have polar groups that can have a proton adducted, like amines, double bonds, or a proton subtracted, acids, hydroxyls, all of these types of molecules can be ionized easily and analyzed by mass spectrometry. That's basically most biological molecules. So the problem with ESI and MS is that actually both of those suffer from interference. People call this commonly matrix effects, but basically the concentration of the other ions, especially if they're high, will interfere with the measurements. So what people have done is basically they introduce liquid chromatography to separate components. The longer the gradient, the longer the column in the LC, the more separated the components are. And that makes the job of the ESI and the MS a little bit easier. In general, there's two main modes of chromatography in modern LC-MS. There's hydrophobic and hydrophilic. Most commonly C18, there's like an alkyl chain that the hydrophobic analytes bind to, and then for hydrophilic, it's the opposite. There's a polar stationary phase, and the hydrophilic molecules bind to that in the LC. So using just these core concepts, you can generate raw LC-MS1 data, right? This is what it actually looks like. You have time, so the scans are happening multiple times a second. You get a mass, an intensity, and then a polarity, because there's a positive and negative scanning mode. So this chromatogram is basically all of the ion intensities at a given time, and you sum them up, and you can see different peaks. These are different molecules, intense molecules. Actually, each one of these time slices is a mass spectrum, and then if you plot it a different way in a mass spectrogram, you can see LC time on the x-axis, mass to charge on the y-axis. You can see all of these little specks are different molecules. So it's quite complex. Quantification is pretty simple. You just take a slice of that spectrogram for a specific mass, say hexose. This is basically a glucose-like mass plus an ammonium ion, and you can plot it. These are the peaks. It's kind of like HPLC. You can cut this into different peaks, sum up those intensities, and that's proportional to the concentration in the solution as it was eluting off of the column. The problem is these are all the same mass, right? You don't know which one's glucose, which one's another hexose, and actually it's not even clear if these are actually hexoses. They just share the same intact mass. So what do we do for identification? Basically, this is what people know as MS-MS or MS2. You can tell the instrument to manipulate the ions and select a particular mass. Just zoom into 198, those hexoses, and you can blow them up. You can crash them into neutral gas molecules. They form a specific set of fragments depending on the chemistry of that molecule. And because those fragmentation patterns are very unique fingerprints, you can match them. So here's an example of serine. It forms different fragments based on what's stable and which bonds are weakest. And typically, there's libraries available that have tens of thousands of molecules that have been purified and analyzed in this way, and you can just match it to your experimental results. You get a pretty good identification if the match score is high. For lipids, it's a little bit different because we have understanding of what the structure is. You can just theoretically predict what those fragments will be. So you can make a giant theoretical library, match all of your lipid MS2 spectra. And for proteomics, it's an additional step, which is you digest the entire protein sequence database for a given species, and then it's kind of like Sanger sequencing. It forms like a mass ladder, right? So you have P, the whole peptide, cut it here, peptide, tide, and so on and so forth. And then you can match these masses. They form unique masses, and then you can get a good fingerprint of what a peptide should be. You match all of your MS2 spectra to all of your theoretical peptides, and then you can basically do protein sequencing. So it's just a little bit of a tangent, but I'd like to also describe a little bit the difference between untargeted and targeted quantification. So in untargeted, we're basically doing the omics. We don't have enough resources, so we're just doing relative quant. We're trying to measure as many things as possible. If you get to the stage where you want to measure something exactly, you'll probably end up doing targeted absolute quantification. This basically means you'll buy a purified standard, so you know exactly what it is. You have perfect positive identification. And then you'll form a standard curve and do your normal quantification. It's expensive, so each one of these is going to cost $500, $1,000. So you can only do a limited number of targeted quants. But you also get the benefit of adjusting for the interferences. So if you spike in an isotope-labeled version of glucose, you can positively identify that that is actually glucose, and it's going to behave exactly the same. So any interferences experienced by this spike-in, like let's say there's a decrease response in the mass spec, then you can basically normalize those out. So that's it. That's mass spec. Hopefully it was relatively simple. Those are all common concepts to proteomics, metabolomics, and lipidomics. Now I'd like to describe a little bit the specifics of each. So you can see these are quite diverse. You have giant 100 kilodalton complexes or beyond, right? You have these small polar molecules, and you have hydrophilic small molecules. The sample prep ends up being quite similar in terms of the actual workflow for lipidomics and metabolomics, except that you're extracting the metabolites with a polar solvent, and then in the lipids, you're extracting with a non-polar solvent. Basically, you extract it from the homogenized tissue or cells, and then you reconstitute in your LC-MS buffer for injection. In contrast, proteomics is quite complicated. You basically have to solubilize the proteins. You have to cleave and cap the disulfides. You digest it with trypsin for a few hours, and then you clean it up before you dry down and reconstitute and analyze. At the LC-MS level, it's quite similar as well. Metabolomics and lipidomics generally use microflow LC, a regular electrospray source. There's positive and negative ions, so some are acids, some are amines. And typically, for a given experiment, you might get around 300 or more metabolites, 1,000 or more lipid species. Proteomics is the odd one out. Basically, the norm is to miniaturize everything to get peak sensitivity. So you're using nanoflow gradients. It's about 1,000-fold lower flow rates compared to microflow. And then nanospray, which is an extremely small needle, they typically only use positive ionization because that works well. And you're getting 2,000 or more, depending on the depth of your setup. You can get up to like 10,000 proteins in a given experiment. And there's an additional level at the data processing where each of the peptides for a given protein are averaged, essentially, and you get the protein quant. So there's a movement recently, and that's to do multiomics, right? So if you're going to go after one, why not go after all of them? In some cases, it's warranted. In some cases, not. But basically, the conventional approach is to do a combined extraction where the polar metabolites are in one phase, the aqueous phase. These are the proteins that are precipitated, and at the bottom you have the lipids, right? And then you basically do 3 different sample preps, 3 different analyses. It gets quite complicated and expensive. Here's the shameless plug for my company. So you might have noticed that all of the instrumentation and sample prep looks quite similar, right? So what Dalton has done is basically invented and developed an assay that does one-shot multiomics. So we will basically keep one sample prep and then one LC-MS analysis. You get all the polar metabolites, the trypsin-derived protein fragments, and then also the lipids in a single run. So you get 3 ohms in one analysis. Okay. In general, the data analysis is quite similar, even with transcriptomics. You're going to do something like a statistical differential analysis comparing cases versus controls. You might do some network analysis where you find correlated groups of molecules, and then you try to learn what their biological functions are, and then you might do some pathway enrichment or gene set enrichment analysis to see if there's any other patterns going on at a higher level. There's other advanced topics that I can't cover here for lack of time, but yeah, if you're interested, we can talk about it at some point later. So what do each of these omics actually offer? So I'll first break down the as a CRO MS analysis service, we basically see a ton of studies. These are the main breakdowns. So typically people are looking at mechanistic studies where they're looking at the biology. They'll sense cells, tissues. There's also biomarker studies, typically serum or some other readily accessible sample. The coverage here is lower, but the benefit is you can sample the animals or people without killing them, right? And then finally, if there's some purified components, let's say you're interested in a mitochondrial phenomenon, then you can send us or send a mass spec core your mitochondrial preps, and they'll analyze that for you. So for proteomics, it's quite similar to RNA-seq. You can get a lot of power or interpretive power from the genetic knowledge base that we've built up as a scientific community, right? So here's one tool that you can find online. You just feed in all of the different genes, and it can tell you the biological processes involved in these top hits, or it can tell you what cell types they were derived from. It can tell you where these proteins are located in the cell, which organelles, what diseases or phenotypes it might be associated with in other studies. So it's quite powerful. And importantly, the diseases and phenotypes signatures, this applies to all omics, really. So in proteomics, it's really the genetic information that you benefit from. For metabolites, basically, you're looking at metabolic information. You might be able to track down a specific metabolic perturbance using enrichment analysis. An additional complementary value is basically looking at nutrients or toxicants. So these aren't really accessible to the animals unless they're fed these nutrients, right? So it's almost like a very easy to modify intervention. In contrast, if you found a protein that you needed to intervene on, I can imagine it'd be like a genetic manipulation or selective breeding. It's quite complicated, right? Finally, lipidomics is relatively underdeveloped, but you can learn stuff about the membrane biology, so whether it's rigid, flexible, thick or thin. There's also lipid-mediated signaling that you might be familiar with, like PIP. You can get some sense of the compartments similar to proteins. Importantly, there's a lot of oxidative stress in the cells, and those attack the unsaturated lipids. You can get a sense of that. And then you can also see some cellular energetic patterns, like whether fatty acids are being metabolized. So these are just some hypothetical study results that you might get if you studied poultry using these different approaches. So, if you looked at woody breast syndrome using proteomics, you might find that some extracellular proteins are disturbed or perturbed, right? And it might be that if you look at the metabolomics, you're finding that a potential cause of that is basically a switching to a glycolytic metabolic activity. And then it might also be related to changes in lipid composition, changing the fluidity of the membranes. Similarly, you can find complementary information in each ohm for a given research topic. So it's the point here is basically each ohm offers unique complementary information that you couldn't really get in any other ohm. So hopefully you have a better idea of what each ohm represents and what it can give for your research program. I think across all of my experience, it's really about answering the right question with the right tool, right? You don't want to use a hammer and just smash everything with RNA-seq when it could be that you really should pay attention to metabolites for a particular problem. Another thing that I've noticed is basically if you're going to do MS or any bioinformatic or omic analysis, you should really consult with the experts that you're going to be working with up front and early on so that they can help you design the experimental statistical design and then also give you an idea of the necessary power that you need to detect your associations, right? So oftentimes I'll be consulting with a group and they have the samples already collected, the entire experimental design is already set up, and then I'm like, okay, this is a little underpowered, you're probably not going to see what you want. So to avoid that, you should basically keep that in mind and consult early. Finally, when we end up getting the results and giving them back to our collaborators or customers, it tends to be, let's just say you can expect these things, which is it's going to be very large and very complex. it's going to be pretty difficult to analyze, and it's going to require both bioinformatic and domain expertise to really understand what's happening, right? Neither group alone is going to have enough expertise to cover all the ground that's in the results. Ultimately, these are hypothesis-generating tools, right? So there's going to be false positives and false negatives, especially when you're testing so many different things. Even if you do multiple testing control and try to filter out the false positives, it's still a problem. And then also be prepared. You might encounter these two common situations where you have a flood of significant hits and you don't know where to go with it, or you might actually have like a few significant hits. So in all cases, basically, it's just the starting point, and you need to follow up with targeted validation experiments, preferably with orthogonal methods, right? So that's brings me to the end. Hopefully, you all have a better understanding of mass spec and what it can possibly do. And if you have any questions or need any advice, I'll be around.